Hi, I'm Karen Sam. I work at CDS Analytical, and I'm going to talk to you about analysis of microplastics in shellfish using pyrolysis GCMS. Take a look at the graph on the left. This is the growth rate of different commodities, such as steel, cement, aluminum, and polymers, otherwise known as plastics. So this is starting from 1970 going up to 2015. And you can see out of all of those, the growth rate of plastics is much higher. 500 million tons of polymer or plastic was produced in 2020. And this is projected to reach 1 billion tons by 2050. So the speed of production of these polymers or plastics keep ramping up. But the problem is we don't have a good way of completely recycling, and it just builds up in the environment. So if we look at the graph on the right, you can see only half of the plastics out there in the environment, only half is either recycled or incinerated. Some of it is still under use, but a lot of it, over half, is simply waste. So this does build up in the environment. So when you think about that, the recycling rate is under 50% globally. This includes both recycling and incinerating. This leaves 50% as waste. And unfortunately, this 8 million tons a year of plastic waste ends up leaking into our oceans. It's also projected by 2050, the weight of plastic in the ocean will be equal to the weight of fish. So what happens when it builds up into the environment? You can see larger pieces of plastics, micro and mesoparticles, littering our landscape and possibly on beaches. This can cause choking risks to animals. And as they're in the environment, they do degrade some more, and especially when it's in the ocean, because you have reactive atmosphere, such as water, UV, heat, and oxygen. So these larger plastics will degrade into micro-sized particles and nano-sized particles, micro being between 20 micrometers and 5 millimeters, and nano being actually under 5 micrometers. So once we've reached that level of that size of particle, then this starts to enter the food chain and enter food tissues because it's consumed by the animals. It's small enough to enter the system of the animal. And then once that happens, as soon as we eat seafood, we are intaking these micro and nanoplastics. We don't know the health effects of the plastics themselves, but a lot of things are attached to the plastics, such as additives, and specifically phthalates. These already have known health risks. So it's really important for us to understand how much plastic is getting into the environment, how much is getting into our food, how much we are consuming, because it's a problem we need to solve sooner rather than later. So how do we do this? How do we determine the profile of plastics in our bodies or in animals or in the environment? There are count-based optical methods such as FTIR, LDIR, and Raman. The benefit to that is other than collecting particles, there's little sample prep. You get information on particle size and types of plastics. It's difficult to analyze particles smaller than 20 micrometers, and it can take from hours to days to analyze just one sample. So it can be a bit time consuming. Another way is a weight-based mass spectrometry method called Pyrolysis GCMS. This requires a little bit more sample prep, as well as a prep for a calibration standard, but you can get an amount of certain microplastics. You can get a quantitative result. You will lose information on the size of particles, but there is no size limitation. This is good for studying very small particles, micro to nano. So in the rest of this presentation, we'll be referring to both micro and nanoplastics as microplastics. And it takes about 40 minutes per sample. Normally, you wouldn't expect GCMS to be a good way to study non-volatile materials like polymers, but pyrolysis works by using intense heat, intense enough to break molecular bonds, transforming polymers into a series of volatile fragments, also called pyrolysates, which can then travel to the GCMS for analysis. There are known mechanisms for pyrolysis degradation so that each polymer produces a unique pyrogram fingerprint. For example, on the top, we're looking at polymethyl methacrylates. All methacrylates will basically unzip along the polymer backbone to produce monomer. Polyolefins, for example, polyethylene, will randomly break along the polymer backbone, so you will get monomer, but also dimer and trimer and larger oligomers. Polymers such as polyvinyl chloride have a weak side group chain. That's the carbon-chlorine bond. So this actually will strip off first. You'll get a peak representing the side group, hydrochloric acid, and then you will get a conjugated backbone that has to stabilize by forming aromatics. So you will get no monomer, dimer, trimer. What you will get is a side group peak and aromatics. So how do we do pyrolysis? A CDS pyroprobe uses a resistively heated filament, which can rapidly heat the sample to the set point temperature. There's a specifically designed sample tube, we call it the drop-in sample chamber tube, which precisely aligns the sample to the center of the heated zone where the drop-in sample chamber is calibrated. This platinum filament is wrapped around a quartz chamber, which we call the drop-in sample chamber, and that can quickly heat to the set point. So this rapid heating, along with the precision placement of the sample and the low dead volume of the pathway, leads to really sharp peaks, excellent pyrolysate transfer, and low RSDs. One difficulty which arises with quantification is the challenge of weighing microgram quantities of often an insoluble polymer to create a standard curve. This is overcome by adding the polymer to a solid matrix, such as diatomaceous earth. And in this experiment, we're quantifying microplastics in shellfish, and we use 6 pure polymer standards. These are known microplastics: polymethyl methacrylate, polyvinyl chloride, polystyrene, polypropylene, polyester, and polyethylene. These were all used as the stock standard. 1 milligram of each standard was added to the diatomaceous earth, and this is ground with a cryo-mill to homogenize this. So the cryo-milling conditions first need to be optimized, so we got a proper homogeneous mixture. So the polymer standard in the matrix was added to a liquid nitrogen tank, and a grinding ball was added, and it was immersed in liquid nitrogen for 5 minutes. It was ground for 40 seconds at 65 hertz, and this was repeated 8 times. And when we could do that, and we ran a repeatability study, we got an RSD of under 5%. Compared to a control, if you only grind this 4 times, you get an RSD of 10%. And to the right is an overlay of actually the 4 repetitions. For shellfish samples, sample prep is required to concentrate the sample, removing the tissue. without degrading any of the existing microplastics. So we looked at 4 types of shellfish. This is oysters, clams, Stimson surf clams, and scallops. They were deshelled and then digested with 1 gram was digested with potassium hydroxide. This was digested at 40 degrees C for 24 hours. Then this mixture was filtered onto a quartz filter. Both the filter and the microplastics on the filter were then cryo-milled using that optimized grinding procedure that we talked about in the previous slide. And when it was time for analysis, we took 2 milligrams of this mixture and pyrolyzed it at 700 degrees. Because one microplastic often produces more than one peak, indicator peaks which represent each microplastic must be chosen for identification and calibration. So the polymer standards were also individually pyrolyzed to determine these preliminary indicator peaks. So here we're looking at a mixed microplastic standard. So these indicator peaks were specific to each microplastic and provided a linear relationship between area abundance and amount. Specific quant ions and retention times were used for the calibration. Using the area of each quant ion, a calibration was performed by weighing 5 different levels of sample into the disc tubes between 0.66 milligrams and 2.44 milligrams to get a calibration range of 0.4 micrograms to 16.27 micrograms. So the quantification of the microplastics in the shellfish was performed by applying linear equations generated from each calibration plot. And it was found that these shellfish, the different shellfish, contain mostly polyethylene and polypropylene, exceeding 80% of that total microplastic amount. So you can see polyethylene was in the greatest abundance, between 10 and 15 micrograms per gram of tissue, while polypropylene was under 2 micrograms per gram. So there is a correlation between the amount of plastic in marine organisms and feeding patterns, marine habitats, and nutritional status. So the main terrestrial microplastic does come from polypropylene and polyethylene. Polyethylene usually exceeds 80% of that microplastic amount. In addition, polyethylene has been reported to dominate microplastics found in marine samples. Thanks.